Okay, continuing along here with the endocrine system. So, a little bit more in detail about what's happening here with cell-to-cell signaling. So, animal chemical signals are usually present in low concentrations, so you're not going to have a whole lot of hormone, and we're going to see how they kind of act on cells and act on each other. As opposed to action potentials, which are quick one-and-done responses, a hormone is going to take longer to present, and it's going to have longer-lasting effects. Okay, so for example, once we begin to release testosterone, it's going to have long-lasting, effects in males, right? Same thing with estrogens in females. Those aren't things that are just going to start and then go away. In combination, these neuro signaling, so the action potentials and chemical signals work together. Okay, and a lot of times, especially with the heart, there's neural influences as well as and this is not just the heart, but just one example, but there's many things in the body that have signals between the nervous system and the endocrine system working together. So how do these signals work in terms of when the hormone is actually in contact with cells and when it's produced? So there's 5 major categories of chemical signaling in animals. So there's autocrine, paracrine, and endocrine signals. Okay, so, and if you look at the prefix for these words, auto means, self, para means in the vicinity, and endo means within, and that's going to deal with we're going to see in a second, that's going to have implications as to what these terms mean. Neural signals having to do with the nervous system, neuroendocrine signals having to do with both the nervous system and the endocrine system, okay? And each of the 5 types of chemical signals can function in any of these categories, right? And they're not functionally distinct, so that means that they can work, you'll see paracrine signals and autocrine signals kind of happening at the same time. It has to do, though, how we group these based on where the molecules originate and where they act. So they're not functionally distinct, but distinct in terms of what they do. So we're not going to see, that an autocrine signal is going to have necessarily a different effect than a paracrine, but rather just more structurally what's happening. So here's the 5 categories. So an autocrine signal is a cell that's going to secrete its own hormones, and then the hormones are going to act on the cell itself. Okay. Paracrine signals, the cell is going to produce hormones, and then they're going to act on neighboring cells. Endocrine signals, what we've been kind of talking about, hormones are carried, from cells to the blood to other kind of far away places. Neural signals, with this we talked about last chapter, the neurotransmitters. So acetylcholine is a neural signal between neurons, short distances in that cleft. And then we got neuroendocrine signals, which are actually neurohormones that are going to be released in the same way from the axon terminal, but rather than going to another neuron, these neurotransmitters are going to enter the blood, and we call them neurohormones. And again, acetylcholine can act in different ways. It can act as a neural signal and a neuroendocrine signal. They're not mutually exclusive. So let's look at some examples of these. So autocrine signals affect the same cell that releases them. So a good example of an autocrine signal is cytokines. And what a cytokine is they're going to amplify the response of a cell to a stimulus. So we see cytokines being produced in response to infections. So the best example is something called interleukin-2. And I always think of cytokines as like breadcrumbs. So T cells secrete these cytokines, and it allows for the cells to kind of see them and rapidly divide and produce more T cells when they see the cytokines. Actually, you may have heard of this cytokines recently because we're, seeing it with COVID-19 that your body creates a cytokine storm in response to COVID-19, and that immune response is actually off the charts and causes a lot of problems. So drugs that kind of quell the immune response and stop that cytokine storm have been looked at to help to alleviate some of the deaths associated with COVID. Paracrine signals diffuse locally and act on target cells near the source, so para means around. So like cytokines, the cell-to-cell signals insulin and glucagon act as paracrine signals and also hormones. Okay, they're produced by 2 areas within the pancreas. They act on nearby pancreatic cells to ensure a steady response to changing blood glucose levels. So if one cell produces the insulin and the glucagon, then the neighboring cells will as well. They also act as hormones by releasing the concentration of insulin and glucagon into the blood, and therefore obviously controlling blood glucose, right? So you look to test your insulin by testing those blood sugar levels and glucagon levels. We talked about endocrine signals already. They're ones that are produced by glands, endocrine glands, then carried to distant parts of the body. Neural signals, another term for neurotransmitters. So remember, a neurotransmitter like acetylcholine, muscle contraction, or dopamine or serotonin are going to be produced from one presynaptic cell at its axon terminal and then received by a postsynaptic cell where it's going to bind to the receptor, change the membrane potential, cause a response in the cell. Okay, so this is very fast because action potentials are propagating and causing the release of those neurotransmitters. But the neural signals themselves are also short-lived because they don't spend a lot of time in that synaptic cleft. They're either reabsorbed or broken down by enzymes. Neuroendocrine signals act at a distance. So these are going to be released from neurons. But once they're released, they are then going to enter the blood and actually act as hormones. So they really do bridge the gap, right? They're produced from neurons, but they enter the blood, so literally they are neurohormones. Okay, so a great example of this is antidiuretic hormone, which is produced by the hypothalamus, and then we know already that it acts on the collecting duct and the distal convoluted tubule to regulate the excretion of water in the body. So there's three pathways in which hormones work. Some endocrine cells respond directly to the environment by secreting hormones. In others, information about environmental conditions is gathered by sensory neurons, integrated, and then hormones are released, okay, which is kind of similar to what we've seen in the nervous system. Or the released neurohormones stimulate other cells in the endocrine system to produce hormones, right? So you see a neurohormone to hormone response through these pathways. And it just depends on the needs and how fast something needs to be done. Okay, so here's the endocrine pathway. There's a stimulus. Stimulates those endocrine cells to produce hormones. They go into the blood, and then they act on an effector cell, which elicits a response. Neuroendocrine pathway, stimulus comes. Okay, neurotransmitter is released. Goes to a postsynaptic cell. Neurohormone is released from that cell, enters the blood, goes to the effector cell, elicits a response. Okay. Neuroendocrine to endocrine pathway, we've got stimulus, sensory cell, neurotransmitter release. Postsynaptic cell here is in the central nervous system, releases a neurohormone into the blood. Okay, that neurohormone then enters the cell, which then will trigger an endocrine cell to release hormone into the blood, act on the effector cell, and elicit a response. Okay, and then that response will feed back to stop the production, and you can see that happening in each of these in different places. So it can stop, the inhibition can take place in different places depending on where, what the situation is and whether or not we've gotten back to homeostasis or not. So all three of these pathways are regulated by negative feedback or positive feedback inhibition, and both of these regulate homeostasis. So in this case, the product of the process is going to inhibit its production. So what is positive feedback? So positive feedback is when the product of a process stimulates its production. And I'll talk about what this means in a second, resulting in greater and greater production of that product. So I mentioned this already with respect to oxytocin and childbirth. So oxytocin, a little bit is produced, a little bit of push starts, right? Start of the endocrine contractions. Once that little bit of push starts, it stimulates the body to start rapidly producing more and more oxytocin. So those contractions keep getting greater, more oxytocin, more, until ultimately the baby is born. That's positive feedback. We also see it with blood clotting as well. Once a clot starts, it rapidly keeps forming until it's shut off, and then it no longer forms it anymore when the wound is healed. Okay. More commonly, though, than not, we see negative feedback. So recall that homeostasis is the maintenance of a constant physical and chemical conditions inside the body. And homeostasis relies on three factors. or three components, I should say, and it's usually always described with a balance, like you see here. So when we're in homeostasis, there's a balance, and then sometimes imbalances take place, and it changes the body's homeostasis. So let's, use something simple like temperature. So, your body's comfortable at 98.6, but then all of a sudden it starts to get really chilly, right? And your body temperature drops down a little bit. That's stimulus, produces a change in the variable. Change is detected by receptors, your sensory organs. They then send their information along the afferent pathway to the control center in the brain, okay, which outputs the information via efferent pathways to effectors, okay? These effectors then are going to do something. In this case, let's say shiver. They're going to cause a little bit of muscle contraction, which is going to stimulate heat, which is going to bring you back to homeostasis, okay? So you can think of it almost like a thermostat in your house, right? You can set your thermostat to 72 degrees, and if it gets too hot, the air conditioner kicks on to get back to homeostasis. If it gets too cold, the opposite, the heater kicks on to get it back to homeostasis. So these are just different ways in which homeostasis works. So here's a real-life example, blood sugar feedback. Okay, I believe we talked about this a little bit in the digestive system chapter, but blood glucose level is your normal level. If your blood glucose level gets too high, beta cells of the pancreas release insulin. Okay, insulin is released. Insulin then instructs the liver to take up glucose, store it as glycogen. Body cells take up more glucose. Glucose level declines, and we get back to homeostasis. Let's say blood glucose level is too low. Okay, so the stimulus here being, low blood sugar level. You skip the meal, right? You start to feel very lightheaded. This would happen if you ate too much, but on this side, you're not eating enough. So in this case, the alpha cells stimulate glucagon. Glucagon then stimulates the liver to break down glycogen and release that glucose into the blood, bringing it back up to a normal level. Okay. So you want to have glucose readily available so that it can then be taken up by the cells. If glucose isn't readily available and taken up by cells, then your liver is going to start breaking down its glycogen reserves for that purpose. ADH, antidiuretic hormones, same kind of thing. In this case, we're talking about water level. Water level is too low, you're dehydrated. Hypothalamus detects that high concentration of solutes, low concentration of water. What does it do? It tells you you're thirsty. Okay, you drink more water. ADH is stimulated to absorb that water. Okay, posterior pituitary produces more ADH in response to that. Let's say water level in the blood here is too high. You're drinking a lot of water. So now you want to produce dilute urine. Hypothalamus detects low solute concentration, releases less ADH or inhibits it, and your kidneys reabsorb less water. You're back to normal. So alcohol is going to inhibit this feedback loop. Blood calcium I talked about earlier, the influence of calcitonin and parathyroid hormone. So blood calcium level at homeostasis. Okay, let's say, we'll start here. Let's say your blood calcium level gets too high. Your thyroid is going to release calcitonin. Calcitonin is going to stimulate blood calcium levels to fall. Okay, what that'll do is it'll stop the breaking down of bone. It'll initiate the osteoblasts to do that, to start building bone. Let's say calcium is too low. Now we need calcium out of that bone, so the PTH is going to be released from the parathyroid hormone, or parathyroid gland is going to release parathyroid hormone, which is going to stimulate your osteoclasts. No, I had that backwards. Your osteoblasts build bone, your osteoclasts break down bone. So here you're going to have the osteoclasts, or maybe I said it right, I don't remember. Your osteoclasts are going to break down bone and put more calcium into the blood. Another very important homeostatic hormone is erythropoietin, and it's involved in blood calcium levels. So when blood oxygen levels fall, the kidneys and other tissues release erythropoietin, which is going to stimulate the production of more red blood cells. More red blood cells mean you can carry more oxygen. Okay, so a lot of times you'll see endurance athletes training in oxygen-deprived environments to get their body to produce more red blood cells, so that way when they're in a regular oxygen environment, they have more than they need, and they can perform better. So I've also seen sometimes people wearing masks, like a special mask when they're exercising in the gym to have that same effect. I don't know how much of an effect that actually does have, though, with the mask. I would assume it has probably a similar effect to the high altitude training. Injections of erythropoietin are illegal, and that's considered blood doping. Lance Armstrong was involved with that, and it's very dangerous because if you really start messing around with stuff like this and you start injecting yourself with EPO, it's going to change your blood viscosity, how thick it is. You can end up with heart attack and stroke. So it's very dangerous. All right, so here's the feedback loop for that. Again, homeostasis, normal blood oxygen. So if there's an imbalance, if it starts to get too much or too little oxygen happening, let's say you're at a really high altitude and you're not getting enough oxygen, or you're even just exercising really hard, your O2 levels in the blood are going to decrease. Your kidney and liver, to a smaller extent, release erythropoietin, which is going to stimulate your red bone marrow to produce more red blood cells, and then red blood cells carry oxygen better. If there's more of them, obviously you're going to carry more oxygen if you've got more red blood cells, and it'll help raise your body's oxygen levels. All right, so that wraps up this chapter on chemical signals in animals. I will see you next time. Make sure you round out the week by finishing up your Mastering Bio assignments and the discussion questions for the week.